discussion is Vibrio, Campylobacter, Helicobacter, Aeromonas, and Plesiomonas. Okay. Okay, so for the learning objectives, at the end of this unit, the student should be able to characterize the significant organisms under Vibrio, Campylobacter, Helicobacter, Aeromonas, and Plesiomonas, describe and explain the clinical significance and pathogenesis of these organisms, and to distinguish features of the organisms mentioned above for accurate laboratory diagnosis. Okay, so first set we have Vibrios. Okay, naman. Okay, so Vibrios are natural habitants of seawater. So they are mostly halophilic with the exception of cholerae and mimicus. They are not halophilic. They are able to tolerate salt, high salt concentration. So they can be called halotolerant. Okay. They can cause infections by drinking contaminated water or eating, or consuming contaminated seafood. Okay. It's isolated mostly from gastrointestinal tract, for patients with gastritis. It can also be isolated from blood and wound infections. The organisms are motile, by virtue of a single polar flagella or flagellum. Okay, they're gram-negative rods, comma-shaped or curved rods. However, in prolonged culture, they may lose that bended shape, okay, and they will retain a straight morphology. Okay, so they could be facultative aerobes, facultative anaerobes rather, or aerobic organisms when cultured. Okay, they are of course oxidase positive except Mechnicoviae, okay, which is oxidase negative, and they are fermentative. When it comes to carbohydrate metabolism, they are fermenters. Okay. So culture media that we use for isolating Vibrios include an enrichment media like alkaline peptone water. Okay, it usually has a pH of about 8.4. So initial isolation, for initial isolation, we use enrichment broth, alkaline peptone water. So we inoculate the organisms there, including the swab. Okay, so if you're going to use the swab is included, okay, inside the tube. Okay, so we incubate it for 5 to 8 hours at 35 degrees Celsius, and then afterwards we can subculture it to TCBS. Okay, so the selective media, that we use for cultivating Vibrios is TCBS, or thiosulfate citrate bile salt sucrose agar. Okay, it contains sucrose and oxgall. Oxgall is a derivative of bile salt. Okay, it also contains sodium citrate and indicators like bromothymol blue and thymol blue. Okay, so the pH is around 8.6. Okay, so the high pH will enhance growth of Vibrios. They cannot be grown in an environment that's acidic. They are killed by acids. Okay, so the high pH, the oxgall, the citrate are important in inhibiting gram-positive bacteria and lactose-fermenting gram-negative bacteria. Okay, sucrose, of course, is added to show or demonstrate sucrose fermentation by the organism. Okay, and if they are able to ferment sucrose, they will produce smooth yellow colonies on the green TCBS. Okay. So this is a table here showing the different species of Vibrio, okay, those that are sucrose positive and those that are not able to ferment sucrose. Okay. So first species, so we'll only be discussing the clinically significant species for this group. we have Vibrio cholerae, no, the representative organism. It is the causative agent of cholera. Okay, so cholera is a gastrointestinal tract infection characterized by severe diarrhea. Okay, death may occur as a result of severe dehydration. Okay, so mode of transmission, of course, ingestion of contaminated seafoods or undercooked shellfish or drinking contaminated water. Okay, Vibrio cholerae again is non-halophilic, but it's halotolerant and it can ferment sucrose. Okay, so antigenic structure and biologic classification, it possesses a single heat labile flagellar antigen. So H is the symbol for flagellar antigen. Okay, so they only have one, but it's poorly immunogenic. Okay, they also possess somatic or O antigen, which is serologically specific. No? There are about 130 to 139 serogroups of the O antigen. Okay, so there may be different O antigens, but they all share the same H antigen. They only have one of that. Okay, the O1 strain is the most common cause of epidemics and pandemics of cholera. Okay, while the non-O1, so the rest of the 129 serogroups are only able to produce cholera-like diseases. Okay, so the Vibrio cholerae serogroup O1 has serotype O1 has three serogroups. So meron tayo nung Ogawa, which has been isolated in India, Inaba from the Philippines, and Hikojima from Japan. Okay, so again, there are more than 130 or 139 serogroups based on typing for the O antigen. Okay, so yan. Okay, so the O1 antigen can be categorized into two biotypes. We have the classical biotype and the L-Tor biotype of the O1, okay, serotype. Okay, so to differentiate them, okay, you can perform RBC hemolysis. Okay, so the classic strain Okay, so the classical strain of O1 is responsible for pandemics in the past. Okay, they are non-hemolytic on blood agar plate, Voges-Prescauer negative, polymyxin B susceptible, and they cannot agglutinate chicken RBCs. Okay, whereas the L-Tor strain is responsible for the recent pandemics. These are more aggressive. No, they are beta hemolytic. They are Voges-Prescauer positive. They are resistant to polymyxin B, no? And they are more resistant to environmental changes, and they can also agglutinate chicken red blood cells. Okay. Now, apart from O1, the L-Tor biotype, the Vibrio cholerae O139, which is probably the last, okay, for the O antigen 139. Okay, this is said to have a similar range of disease with the Vibrio cholerae O1 L-Tor biotype. Now, to differentiate, you can perform antigen detection, no? So you detect O1 antigen or detect the polysaccharide capsules. So O1 antigen is positive for, of course, Vibrio cholerae O1, and the polysaccharide capsule is present for O139. Okay, so that's how we differentiate O139 and O1 L-Tor biotype because they are said to be similar. Okay. Oh, let me go back to that pala. Okay, so look, okay, so the classical type is responsible for the first 6 pandemics that we have had, no? 1817, 1829, 1846, 1863, 1881, and 1899. Okay, so nagrabi-grabi diri sa pula. Okay. However, for the 7th pandemic in 1961, that has been caused by the L-Tor type. Okay. So for the pathology and pathogenesis, okay, so our Vibrio cholerae is pathogenic only for humans. Okay, under normal gastric acidity, an individual would, for him to be infected, okay, more than 10 to the 10th power of Vibrio cholerae or more has to be present in water, for infection to occur. Okay, in food, less than, so about less than 10 is to 2 up to 10 is to 4th power of the organisms in food, okay, is enough to cause infection. So kailangan mas marami sa water, okay, to cause infection. That's under normal gastric acidity. Okay, now if the patient is taking acid suppressors, then a small amount may lead to infections of Vibrio cholerae. Okay. Now, the pathogenesis of the infection is as follows. It can produce severe diarrhea. It can also cause mild infections. Okay. The severe type of infection is due to the presence of a potent enterotoxin called cholerae. Okay. This enterotoxin affects the buccal bowel mucosa, okay, which leads to outpouring of water and electrolytes, hence the characteristic rice watery stool, basa ang tae. No? Yun yung effect ng cholerae. Okay? So this would lead to severe dehydration and severe muscle cramping and anuria. Kasi sige-sige ang pupo, no? Ang bowel movement. Okay? So severe dehydration, of course, may lead to hypovolemic shock and circulatory collapse, and eventually would lead to death. Okay? So in mild infections, individuals may be asymptomatic or the diarrhea may be self-limiting. Okay? So the individual may be a carrier. Okay? So Vibrio cholerae usually produces heat labile enterotoxin with the A and B subunits. So yung type ng enterotoxin niya yung may A and B subunits. Its A subunit causes an increased level of cyclic AMP. Okay, so an increase in cyclic AMP levels usually leads to hypersecretion of water and electrolytes into the lumen. Okay, kaya lumalabas lahat. Okay, the diarrhea usually around 20 to 30 liters of water. Okay, I think this is a day. Okay, so grabe kadaghan, no, na fluids na lumalabas. So this would lead, of course, to dehydration. Kaya dapat paspas din ang fluid and electrolyte replacement. Dahil pag hindi, it would lead to, of course, shock, no, acidosis, and death. Okay, so the diagnosis for the laboratory portion can be done by the usual, no? So microscopy. Okay, of course, the specimen is stool. No? Observe for the characteristic rice watery stool ng pasyente. Rectal swab may be accepted usually in the acute phase or acute stage. Specimen transport may be done with the use of Carey-Blair. Kung kailangan i-transport, then we use Carey-Blair. Okay? So gram staining would result to gram-negative comma-shaped rods. Okay? Motility may be best observed using dark field or phase contrast microscopes. Okay? For culture, we can grow them on peptone agar. No? They can grow rapidly on peptone agar. And then we can also grow them on blood agar plate with a pH 9. And then TCBS, of course. No? So on TCBS, they grow smooth yellow colonies. Okay? Yung ating ito, like this. Okay? Smooth yellow colonies. Okay? Now, to identify, we can also perform string test using 0.5% sodium deoxycholate. And this is added to the organism. Okay, mix it and then observe for the production of a viscous string, this one. Okay, so this one is an indication of Vibrio cholerae presence. And then, of course, serotyping should be done to differentiate serogroups O1 and O139, especially during times of cholera epidemics. And we can also perform and observe biochemical reaction patterns. Okay, now for treatment, water and electrolyte replacement is really, really important. No? Oral tetracycline may be given. However, note that some Vibrio cholerae may have already acquired assistance because of transmissible plasmids, no? Yung mga jumping genes na yan. And of course, again, perform sensitivity testing or susceptibility testing. Okay, so this table here shows you the different species of Vibrio, okay, and the tests that you can use to differentiate them. Okay, so Vibrio cholerae is non-halophilic, like Mimicus. For sucrose fermentation, cholerae is positive. Okay, Mimicus is negative. For ADH, positive lang si Fluvialis and Furnacii. Okay, for Cellubios vulnificus, for Voges prescauer, Anginolyticus. Okay. So Vibrio cholerae is the agent of epidemic and pandemic cholera. Now the so-called Asiatic cholera, which was first noted in India. Okay, because of their culture and practices. Okay. And then note, no, Vibrio parahemolyticus, which is the most common agent of infections caused by eating contaminated seafoods like oysters. No? So it is able to demonstrate a phenomenon wherein the strains isolated from human hosts or the clinical isolates demonstrate hemolytic capacities on blood agar plate, okay? While those isolated from non-human sources are not hemolytic, okay? This is observable in a special culture media called the Wagatsuma agar. Okay, so the phenomenon described is Kanagawa phenomenon. This is possible because of the presence of a thermostable hemolysin called the Kanagawa toxin, which is seen only in human samples. Okay, so Kanagawa phenomenon is demonstrated by Vibrio parahemolyticus from specimen isolated from humans. Okay, and this can be demonstrated best in the Wagatsuma agar. The Wagatsuma agar contains, it's a blood agar plate, no? Wagatsuma blood agar plate. It contains high salt mannitol concentration. Okay, so it has mannitol and high salt concentration. Okay, let's move on to Aeromonas. Okay, so Aeromonas is found in both fresh water and seawater. It can cause gastroenteritis, cellulitis, wound infections in humans, no? After coming into contact with contaminated water, either from the drain, the sink, or tap water, and even sometimes distilled water. Okay, but they are also known to cause diseases in cold-blooded animals. Yeah, merong Aeromonas infection sa fish. Okay, lab diagnosis, we can grow them on MAC, on EMB, on SSA. No, so these are same culture media that we use for identification of enteric organisms. So we have to perform oxidase tests. No? We can also grow them on specialized media like the SYN medium, no, remembers? Cefsoludin, Irgasan, Novobiocin medium. Okay, so we can also perform fermentation of glucose and indole production, okay, to differentiate them from Pseudomonas. Okay, they are motile by virtue of a single polar flagella. Okay, the most common human isolate is Aeromonas hydrophilia. Okay, so hydrophilia means water loving. Okay, Okay, so descriptions or characteristics rather, they're able to produce heat-labile enterotoxin and heat-stable cytotoxic enterotoxin. Okay, so may heat-labile enterotoxin at meron din silang heat-stable cytotoxic enterotoxin. They're also able to produce enzymes like protease, lipase, and nuclease. Okay. Then we have Plesiomonas. No? So microscopically, they're motile, straight, ends are rounded. No? Of course, you have negative bacilli. Previously, they belong to the Vibrionaceae family together with Aeromonas. But currently, they're categorized as enteric bacteria. Hence, during your discussion, kasama na si Plesiomonas. Okay, so there's only one clinically significant species for this, and that's Shigeloides. It's important clinically in Japan, and in tropical and subtropical areas also as agent of gastroenteritis. The infection caused by this organism is mild with no blood or mucus in stool. Of course, again, paano nakukuha? Ingestion lang ng maduming pagkain. So ingat-ingat. Ito talaga ang nagdadala ng problema, yung maduming pagkain. Okay? So lab diagnosis, we can grow them on blood agar plate. We can also grow them on MAC and EMB. Of course, they're non-lactose fermenters on MAC and EMB. Okay, they are oxidase and indole positive, to differentiate them from other species of enteric organism. Ayan. So this table shows you the differences between the three members, previous members of Vibrionaceae. So again, as mentioned, this is currently a member already of the enteric group. Okay, so let's move on to the next organism. We have Campylobacter. Okay, so Campylobacter jejuni and Campylobacter cholerae are most frequent isolated species of Campylobacter in humans. Now, technically, they are clinically indistinguishable. Both of them are curved, gram-negative bacilli. But for better differentiation, okay, it is said that it is best to substitute carboflexin as the counter stain when you perform gram staining. So remember gram staining, the counter stain uses safranin. However, when your organism is Campylobacter, the differentiation is improved when you change safranin into carboflexin. Okay, so there are 19 species of Campylobacter, but the most clinically significant is jejuni, which is the agent of most human infections, followed by Campylobacter cholerae, which causes about 5 to 10% of infections in humans. Okay, so again, they are both microaerophilic, capnophilic, catalase and oxidase positive. Both are non-oxidizers. Okay, they cannot oxidize or ferment carbohydrates. Okay, so green yung dalawang nilang OF media. Okay, they use amino acids as their source of energy. Okay, so they're gram-negative rods. They may show comma-shaped or S-shaped or gull-winged shapes. Mukha daw seagull like this. Ito yung wings ng seagull. At ito naman yung organism. Ayan, so gull-winged shapes. They're able to cause gastroenteritis and diarrhea. And they have been associated in the Guillain-Barré syndrome. Guillain-Barré syndrome. It is an autoimmune disorder resulting from cross-reactivity of Campylobacter antibodies with the nerve ganglia. Okay, so ang mode of transmission is ingestion again of contaminated materials and direct contact. Okay, so direct contact with contaminated animal feces or direct contact with infected person. Okay, animal carriers for Campylobacter are the usual farm animals. Okay, it has a characteristic darting motility, parang mabilis yung movement. No? This particular motility is best demonstrated when using dark field microscope. And so they are motile with single polar flagellum. Okay? So pathogenesis in ang ating Campylobacter. Okay? So it is able to produce cytotoxins, cytotoxic factors, and enterotoxin. The organism is believed to multiply in the small intestine, and then from there they could invade the epithelium, which would lead to inflammation. Eventually, this results in the appearance of blood cells in stool. Okay. Sometimes the bloodstream is invaded and it could cause endocarditis, septic arthritis, and even meningitis. Okay, so to diagnose the presence of Campylobacter, stool would be the best sample. Okay, of course, gram stains may show gull wing shaped rods that are gram negative. For cultivation, it is best to use selective media. So we have Campylobacter blood agar. Okay, so this blood agar is selective due to the presence of vancomycin, polymyxin B, cephalothrin, trimethoprim, and amphotericin B. Sciurus medium is quite similar, but it only contains vancomycin, polymyxin B, and trimethoprim. Okay, the organism is quite difficult to isolate because it needs special conditions for growth. No, medyo OA ito, fastidious. It is incubated at reduced atmospheric oxygen. Okay, so about 5% oxygen lang, with 10% carbon dioxide and 85% nitrogen. The optimum temperature for growth is usually at 42 to 43 degrees Celsius. Okay, so this temperature usually prevents growth of enteric organisms. Patay ng enteric organisms sa 42 to 42 degrees. Okay, growth is usually observed within 48 hours. Okay, the colonies may be gray to pink or yellow to gray colonies. They are, of course, non-hemolytic, and it may be slightly mucoid. Okay, so these are the characteristic species, no, jejuni, coli, and fetus, pero not as common as the first two. Okay, so at 25 degrees Celsius, fetus shows growth. At 37 degrees Celsius, all three species are positive. At 42 degrees, only jejuni and coli can grow. However, to differentiate jejuni and coli, you can perform hypurate hydrolysis. Jejuni is positive for hypurate hydrolysis. For nalidixic acid, okay, both jejuni and coli are susceptible. And for cephalotin, both jejuni and coli are resistant. Okay. Next, we have Helicobacter organism. So previously, they belong to Campylobacter. So previous name is Campylobacter pylori. So there's only one clinically significant species for this, and that's Helicobacter pylori. The characteristic is that it's small, no, mahirap hanapin microscopically, true, no, curved gram-negative bacilli. Sometimes they only demonstrate the U form. It's microaerophilic, like Campylobacter. Okay, it would require 5% oxygen with 10% carbon dioxide and 85% nitrogen, and it should be incubated at 35 to 37 degrees. It cannot be grown at 42 degrees Celsius. Initra. Okay, it's motile by virtue of a tuft of flagella at the end. Ayan, Solophotricus ito, si Helicobacter. Okay. It's a very, very strong producer of urease. Okay? So it's oxidase and catalase positive, but it cannot reduce nitrate to nitrite. Okay, so for pathogenesis ng ating Helicobacter pylori, okay, so the natural habitat of our organism is the mucus-secreting cells of the stomach. Okay, this organism is known to be the agent of antral gastritis, duodenal ulcers, peptic ulcers, gastric ulcers, and it has also been implicated in gastric adenocarcinoma. Okay, this organism grows best at pH 6 to 7. It cannot actually survive the acidic pH of the gastric lumen, so it burrows itself into the folds of the stomach wall. Remember, yung inner wall ng stomach natin has folds. It hides itself there. Okay, so it invades the mucosa. Okay, and then it releases the toxins and lipopolysaccharides. It damages the mucosal cells. Okay? So it produces proteases and ureases and cause damage to the mucus secreting cells. So when these cells are damaged, they can no longer produce mucus. Okay? Remember, mucus is very important, covering of this epithelium to protect it from the effects of the acids found inside the lumen. Okay. Now, if there are damaged mucus secreting cells, okay, the amount of mucus covering the lining might lessen, and this may cause the acids to damage the mucus cells further, okay, producing lesions or ulcers. Okay. The presence of the large amounts of urease will produce large amounts of ammonia, and this is also detrimental to the naturally acidic environment. So the large amounts of ammonia produced from the activity of bacterial urease, it will neutralize the hydrochloric acid, which is bad for the functioning of our stomach. Okay. So clinical syndromes. Okay. Again, it is implicated usually in chronic gastritis, in duodenal ulcers, peptic ulcers. Ayan, no? So nagbubutas-butas. Okay. Normally, the lining of the stomach is wrapped in mucus, okay, due to the mucus released by the mucus-secreting glands, the mucus-secreting cells. However, if the mucus-secreting cells are damaged because of the action and activity of H. pylori, okay, the mucus protecting that wall will lessen, okay, causing the acids to penetrate into the lining, producing these ulcerations. Okay. So gastric biopsy for histologic examination would be best to identify Helicobacter pylori. Okay. So for a laboratory identification of H. pylori, okay, we can perform special silver stain or modified GEMSA. Okay. The gold standard is still histological staining, okay, and of course culture of biopsied tissue from the stomach or the duodenum. Okay, so what do we do with the tissue? We mash it or ground it, and then take a sample of that mashed tissue and plate it on ship's blood agar, incubate it at 37 degrees Celsius under microaerophilic conditions and increased humidity. Yeah. Eventually, colonies would grow, showing small, gray, translucent characteristics that are slightly beta hemolytic. Okay. Biochemical testing can also be done. Okay. Remember, they are able to strongly produce and rapidly react, producing the positive cholerae reaction that we see in biochemical testing. Okay, so they are strong producers of urease. Okay. So diagnosis, apart from cultivation, biochemical test, culture, we can also identify them using serologic methods. We can determine serum IgG antibodies for H. pylori. We can perform stool antigen tests via ELISA. We can also perform carbon urea breath test, which detects urease activity. Okay, so this is usually done to confirm Helicobacter colonization and to monitor the treatment for H. pylori. Okay, so breath test is commonly done for that. Now, endoscopy is also done to in gathering samples, for performing biopsy, collecting specimen for testing, and microbial culture. Okay, so for treatment, like those previously mentioned, it's best to perform or to give triple therapy, metronidazole or bismuth subsalicylate or bismuth subcitrate plus amoxicillin or tetracycline. So tatlo, metronidazole, and then a choice between bismuth subsalicylate or bismuth subcitrate plus amoxicillin or tetracycline. So that treatment is given for 2 weeks. Okay, so aside from that, we can provide acid suppressing agents and proton pump inhibitor. Both will lessen the acidity of the environment and help the ulcers heal. Okay, so this is usually given for 4 to 6 weeks. Okay, so this is a table showing the differences between the species of Campylobacter and the Helicobacter species pylori. Okay, so to differentiate, jejuni and coli perform high purate hydrolysis, positive si jejuni, negative si coli. Okay, what else? Fetus is positive for growth at 25 degrees Celsius and susceptible to cephalotin. Helicobacter pylori is easily distinguishable by a strongly positive production of urease enzyme, and it is resistant to nalidixic acid. Okay, so key points before we end this discussion. So the key points is that most species in this group are found in fresh or estuarine or marine waters. Vibrio cholerae produces a powerful enterotoxin, choleragen, and is responsible for large numbers of epidemics and pandemics. Okay? Other Vibrio species are common causes of diarrheal diseases, usually related to consumption of raw shellfish or related to aquatic wound infections. Okay? TCBS, of course, is the agar or the medium of choice to isolate and differentiate Vibrio species. Okay? Aeromonas species can cause infections that may lead to septicemia, meningitis, and even death. Okay? So, Campylobacter naman are generally responsible for enteritis. So, jejuni is one of the most common causes of bacterial diarrhea worldwide. It has a characteristic seagull wing shape or morphology under the microscope. And it is associated usually with the infection or with the syndrome Guillain-Barré syndrome. Okay, so patients that are positive with this syndrome are almost always positive for Campylobacter antibodies. Okay? And then H. pylori is strongly associated with gastric and duodenal ulcers. So it is able to cause the production of such infections due to its activity. It is able to produce urease, which produces ammonia, which affects the acidity of the gastric environment. Okay? And it has also been implicated in gastric cancers or gastric carcinoma. Okay, so that's the end of today's discussion. Thank you for listening.